Hey guys, in this video tutorial, we'll be learning about JAk-STAT pathway. Jak is the name of a Greek god having two faces like this receptor, and STAT, it is a signal transducer and activator of transcription protein. In the JAK-STAT pathway, before the ligand binding to the receptor are present as monomers. After the ligand bind to the receptor dimerization takes place. The ligand can be cytokine, growth factor, interleukin, interferon, granulocyte, macrophage colony stimulating factor, leptin, and prolactin. After the binding of ligand to the receptor, the cytosolic domain Janus kinase or JAK domain activated by cross phosphorylation. In this process, the JAK, the JAK domain cross phosphorylate each other. After the cross phosphorylation, the Janus kinase phosphorylate the receptor at cytosolic domain, and after that the phosphorylated JAK domain phosphorylate the STAT protein. The STAT protein phosphorylate and form dimer. If the two STAT protein are same, then it is homodimer. If the two STAT protein are different, then it is heterodimer. After the STAT protein activate different transcription factor. If we will see the different structure of STAT protein, different domain of STAT protein, this can be like this. It has one N-terminal domain attached to NH2, which help in dimer formation. This dimer formation, and near to this there is a coiled domain which act as NLS or nucleus localization signal, which guide this SH STAT dimer to the nucleus. And there is a DNA binding domain which bind to the DNA that need to be transcribed to RNA, and a SH2 domain which dock to this Janus kinase protein, and a C-termini transactivator which help in the gene expression. The STAT protein after transcription, it can activate different processes, like it activate the BCL2 and XL, which lead to anti-apoptosis of the cell. Like that, it also activates cis or SOX that lead to apoptosis of cell. That also form c-MYC, which help in proliferation or cell cycle, and it also activate P21, which downregulate the cell cycle. It also form activates it also activate AOX transcription factor, which help in lipid metabolism. The Janus kinase also activate the AKT or ERK pathway in its downstream. As it has tyrosine residue phosphorylated at the cytosolic domain, so the JAk-STAT pathway also known as RTK or receptor tyrosine kinase pathway. I will request you to repeat the video and try to solve the questions. So this is a question from CSIR net December 2017. In this question, this is giving some data about receptor tyrosine kinase and it's asking which of the following option the statement given regarding RTK is incorrect. So the option A is all RTK have three essential components: an extracellular domain containing ligand binding site, a transmembrane domain, and a cytoplasmic segment that includes a domain with protein tyrosine kinase. This statement is exactly there is three domain: outer, transmembrane, and intracellular. Option B is most RTK are monomeric and ligand binding to extracellular domain induces the formation of receptor dimer. Exactly, the monomeric receptor become dimer after binding with the ligand. The option C is all cytokine receptor belong to RTK. No, not all. Some of the cytokine like IL-1, this cytokine receptor belong to RTK, but not all. Some of the cytokine receptor belong to GPCR also. So the option C is wrong. The option D given is the ligand binding to RTK lead to autophosphorylation of the protein tyrosine kinase in the cytoplasmic domain. The activated kinase then phosphorylate several tyrosine residue in the receptor cytoplasmic domain. Immediately after the binding of the ligand, it autophosphorylate at a tyrosine residue and the receptor cytoplasmic domain become activated. So the answer for this question is C. So in this question it's asking the RTK protein contain all the following structural feature except an extracellular ligand binding domain. Exactly, it has the receptor monomeric receptor and after ligand binding it become dimer. So this is extracellular ligand binding domain. A cytosolic phosphatase domain. No. It has a cytosolic kinase domain which phosphorylate the SH2 protein. A cytosolic kinase domain, it's correct, and a dimerization domain. The two receptor are also known as dimerization domain. In this question it's given that cell surface receptor that following ligand binding can phosphorylate tyrosine residue on themselves on cytoplasmic substrate. They are involved primarily in control of cell growth and differentiation. It's clearly mentioned that the ligand can phosphorylate tyrosine residue and it's involving in cell growth. So the answer is receptor tyrosine kinase.